Hello there, and welcome to Biology for Sleep, a place where science slows down, curiosity becomes gentle, and learning is allowed to feel unhurried. This is not a space built for urgency. It does not ask you to be alert, productive, or efficient. It exists for the hours when your body is ready to rest but your mind is still moving, when the day has left its marks in the form of lingering thoughts, half-finished worries, and small loops of attention that keep returning to the same places. Here, the goal is not to stimulate or impress. The goal is to explain quietly, steadily, in a way that gives your thoughts something soft to settle into. Tonight's subject is dopamine, a small chemical that carries an outsized influence over how life feels from the inside. It shapes what draws your attention, what feels worth doing, and what quietly fades into the background. It influences motivation, learning, habit, anticipation, and the subtle sense of direction that guides behavior long before conscious thought arrives. Dopamine does not dictate your actions in a mechanical way. It does not override choice, but it does prepare the ground on which choice takes place. It tilts the landscape. It makes some paths feel easier to step onto than others. In popular culture, dopamine is often reduced to a single phrase. It is called the pleasure chemical. That label sounds tidy, but it is misleading. Dopamine is not a direct generator of happiness. It is not a switch that turns joy on and off. It is closer to a signaling system that tells the brain what is relevant right now and what might be worth pursuing next. It is active not only when something feels good, but when something seems important. It rises when something is novel. It responds to expectation. It becomes active when there is a gap between where you are and where you might go. Every moment your brain is sorting information. Sounds, images, memories, plans, and bodily sensations are all competing for limited processing space. Most of this information never reaches conscious awareness. It is filtered, prioritized, or ignored before it becomes a thought. Dopamine participates in that filtering. When dopamine rises in certain circuits, the brain marks something as significant. It becomes easier to notice, easier to remember, easier to act upon. When dopamine is quiet, things still exist, but they lose their pull. This system evolved long before screens, schedules, or written language. It developed in nervous systems that needed to decide quickly what mattered in environments filled with uncertainty. A change in sound could signal danger. A new shape in the distance could mean food. Dopamine helped organisms orient toward what might alter survival. It did not care about long-term fulfillment. It cared about relevance. It answered a simple internal problem. Out of everything happening, what deserves energy right now? That same system now operates in a world that delivers novelty on demand. A glowing screen can present something new every second. Each new image, message, or headline triggers small shifts in the same circuits that once responded to movement in tall grass or the sight of ripe fruit. The brain does not distinguish between ancient relevance and modern stimulation. It responds to change, contrast, and unpredictability. Dopamine rises not because something is meaningful, but because it is different. This is not a flaw. It is a feature that evolved in a very different environment. The nervous system you carry was not designed for constant input. It was designed for a world where change was informative and rare. In that older world, a surge of dopamine pointed towards something that might matter. In the modern world, that signal can be activated by nearly anything engineered to capture attention. Understanding dopamine is not about blaming yourself for being distracted or driven. It is about recognizing the structure of the system you inhabit. The brain does not ask whether a stimulus is healthy. It asks whether it is salient. It does not ask whether something will be satisfying in the long run. It asks whether it stands out now. This has consequences that unfold quietly across a lifetime. It shapes how habits form. It influences how effort feels. It affects the way learning sticks or slides away. It changes how boredom feels in the body. It plays a role in addiction, not as a moral failing, but but as a predictable outcome of circuits built to repeat what once seemed important. It participates in mood, not as the sole driver, but as part of a network that links expectation, movement, and reward. Dopamine is also deeply involved in how time feels. It is more active when you anticipate than when you arrive. It rises before action more than after completion. It is involved in the stretch of waiting and the pull of wanting. It shapes the difference between having and hoping. This means that much of what feels like personality is influenced by chemistry. Some people experience stronger signals in these circuits. Some have systems that respond more intensely to novelty. Some have baselines that run higher or lower. These differences are shaped by genes, development, experience, sleep, stress, and environment. They are not chosen. They are inherited and learned in ways that begin long before conscious memory. This does not make identity chemical. It makes identity embodied. The mind is not separate from the brain. Thought is not separate from signal. Motivation is not separate from movement in cells. Every idea you have ever had occurred in tissue that depends on molecules. That does not reduce meaning. It grounds it. If this kind of exploration feels calming or helpful, supporting this space allows it to reach others who are also searching for something quieter than the usual rush. This channel grows slowly, intentionally. It is meant to be a place where learning does not feel like pressure. Over the course of this series, dopamine will be followed through its many roles. Its pathways in the brain will be traced in plain language. Its role in habit will be examined without judgment. Its interaction with stress and sleep will be explained in concrete terms. Its changes across the lifespan will be described with attention to both biology and experience. The effects of technology will be approached as a design problem rather than a personal failure. This is not about self-improvement. It is not about control. It is about clarity. Dopamine does not command behavior. It influences probability. It makes some actions feel heavier and others lighter. It does not remove choice. It shapes the field in which choice occurs. When a hill is steep, walking requires effort. When the ground slopes gently, movement happens almost without notice. Dopamine creates those slopes in the mind. Most of this happens beneath awareness. You do not feel dopamine directly. You feel its consequences. You feel interest. You feel restlessness. You feel pull. You feel the vague sense that something is missing or that something else would be better. These feelings are not mistakes. They are outputs of a system designed to keep organisms moving through uncertain environments. The modern challenge is not that dopamine exists. The challenge is that the environment now speaks to it constantly, often without regard for long-term balance. The brain responds exactly as it was built to respond. The cost is felt later as fatigue, distraction, dissatisfaction, or a sense that attention no longer stays where it is placed. None of this is a personal failure. It is the predictable result of a nervous system meeting a world it did not evolve to handle. Understanding dopamine offers a different frame. Instead of asking why willpower fails, it asks how circuits are being engaged. Instead of asking why focus is difficult, it asks what the system is being trained to expect. Instead of asking why rest feels hard, it asks how stimulation has shifted the baseline of arousal. These questions do not demand action. They do not prescribe change. They simply describe what is happening. As the pace of thought slows, this becomes a good time to begin tracing that description, not in a dramatic arc, not in a moral tone, in steady, grounded language that respects both the science and the lived experience of having a human brain. The next step is to clarify what dopamine actually is at the biological level, not as a metaphor, not as a headline, but as a molecule that moves between cells, alters electrical patterns, and participates in the communication that makes thought possible. From there, its effects become easier to understand because they are no longer mysterious forces. They are signals travelling along defined pathways shaped by history and environment, doing exactly what they were built to do. And in that clarity, the mind often finds it easier to rest. When people hear the word dopamine, they often picture a bright internal spark, a burst of pleasure, a kind of emotional sugar that the brain releases when something good happens. That image is understandable, because many experiences that feel rewarding are associated with activity in dopamine pathways. But dopamine itself is not a feeling. It is not pleasure. It is not happiness. It is not satisfaction. Dopamine is a neurotransmitter, a small molecule used by neurons to communicate with one another. It is part of the language the brain uses to move information from one cell to the next. Neurons do not touch. They sit separated by microscopic gaps called synapses. When one neuron wants to influence another, it releases chemicals into that gap. These chemicals drift across and bind to receptors on the next cell, changing its electrical state. Dopamine is one of many such messengers. Others include serotonin, norepinephrine, acetylcholine, glutamate, and GABA. Each has its own roles and patterns, its own distribution across the brain, and its own effects on how circuits behave. Dopamine's uniqueness lies not in its chemical structure, but in where it is used and what those circuits do. Most dopamine in the brain is produced in a small number of regions deep in the midbrain. Two of the most important are the ventral tegmental area and the substantia nigra. From these hubs, dopamine-producing neurons send long projections to other parts of the brain. These pathways form networks that link perception, movement, memory, emotion, and decision-making. One major pathway connects the ventral tegmental area to regions involved in motivation and valuation, such as the nucleus accumbens and parts of the prefrontal cortex. This is often called the mesolimbic and mesocortical system. Another pathway connects the substantia nigra to structures involved in movement, particularly the basal ganglia. This is the nigrostriatal pathway, and its degeneration is what leads to the motor symptoms of Parkinson's disease. These pathways illustrate something important. Dopamine is not just about reward. It is deeply involved in movement. It is necessary for initiating action. It influences how easily a motor plan becomes a movement. When dopamine in the nigrostriatal pathway declines, people experience slowness, rigidity, and difficulty starting motions. This is not because they no longer want to move. It is because the chemical support that allows intentions to become actions is impaired. In motivational circuits, dopamine plays a similar role. It does not generate desire in a simple sense. It alters the probability that a thought or impulse will translate into behavior. When dopamine rises in these circuits, the brain becomes more likely to orient towards something. Attention shifts. Energy mobilizes. The idea of acting feels easier. Crucially, dopamine does not spike most strongly when a reward is received. It spikes when a reward is predicted. Early experiments in animals revealed this pattern clearly. When a rat learns that a sound predicts food, dopamine neurons begin to fire in response to the sound, not the food. Once the association is learned, the actual delivery of food produces little additional dopamine activity. If the sound plays and the food does not arrive, dopamine activity drops below baseline. This pattern shows that dopamine is about expectation and learning, not pleasure. It signals a difference between what is predicted and what actually occurs. When something better than expected happens, dopamine increases. When something worse than expected happens, it decreases. This is called a reward prediction error. It is a teaching signal. It updates the brain's model of the world. If you expect nothing and receive something, dopamine rises. If you expect something and receive nothing, it falls. Over time, this mechanism trains the brain to anticipate patterns. It strengthens connections between cues and outcomes. It teaches the nervous system what tends to lead to what. This is how habits form. A cue appears. Dopamine rises slightly in anticipation. An action follows. An outcome occurs. If the outcome matches or exceeds expectation, the circuit strengthens. The next time the cue appears, the response is faster. Eventually, the behavior becomes automatic, triggered by the cue before conscious thought intervenes. This process does not ask whether the habit is good or harmful. It asks whether the pattern is reliable. Dopamine does not encode value in a moral sense. It encodes predictability and salience. If something consistently produces a noticeable outcome, the system learns it. This is why highly engineered stimuli are so effective. They provide frequent, unpredictable outcomes. Each scroll might reveal something interesting. Each click might lead to novelty. The variability itself drives learning. The brain cannot easily settle because the system is designed to stay alert in the face of uncertain reward. This does not mean dopamine makes people compulsive. It means the system is optimized for environments where unpredictability signaled opportunity. In the modern environment, that same mechanism is constantly engaged. Another common misunderstanding is that more dopamine is always better. In reality, both too little and too much dopamine can impair function. The brain operates within ranges. In some circuits, low dopamine leads to apathy and slowness. In others, excessive dopamine can contribute to impulsivity, agitation, or distorted beliefs. Conditions such as schizophrenia and mania involve dysregulation of dopamine systems. The problem is not simply high levels, it is inappropriate signaling in certain pathways. Dopamine that fires at the wrong times or in the wrong contexts can cause the brain to assign importance to irrelevant stimuli. Neutral events begin to feel charged. Random coincidences seem meaningful. The mind starts drawing connections where none exist. This illustrates another key role of dopamine. It participates in the assignment of significance. It helps determine what feels important. When this system misfires, the sense of relevance becomes distorted. In everyday life, this system is usually well regulated. It highlights things that genuinely matter for survival and functioning. It responds to food, social cues, goals, and learning. It supports curiosity. It reinforces skill acquisition. It helps maintain direction over time. Dopamine does not work alone. It interacts constantly with other neurotransmitters and hormones. Serotonin influences mood stability and patience. Norepinephrine affects arousal and vigilance. Cortisol shapes responses to stress. Melatonin regulates sleep. Dopamine sits among these, integrating information about prediction, effort, and movement. This interaction means that dopamine's effects depend on context. In a rested, calm brain, a dopamine signal may feel like interest. In a stressed brain, the same signal may feel like urgency. In a sleep-deprived brain, it may feel jittery or unsatisfying. Chemistry does not operate in isolation. It is also important to distinguish between tonic and phasic dopamine. Tonic dopamine refers to baseline levels. Phasic dopamine refers to brief bursts in response to events. Baseline levels influence overall motivation and energy. Bursts drive learning and adaptation. A brain with low tonic dopamine may experience the world as flat. Tasks feel heavy. Initiation is difficult. A brain with excessively high tonic dopamine may feel restless or overdriven. Balance matters. Phasic bursts teach. They mark moments when expectations are violated. They adjust future behavior. Without these bursts, learning slows. With too many bursts from trivial stimuli, attention fragments. Dopamine is therefore not a pleasure faucet. It is a regulatory signal that modulates how strongly the brain leans toward the next thing. It does not tell you that something is good. It tells your brain that something deserves processing, memory, or action. When people say dopamine controls life, what they often mean is that it shapes the invisible currents beneath choice. It influences what feels worth effort. It biases what stand out. It alters the slope of mental terrain. But it does not remove agency. It provides probabilities. It does not command, it suggests. Understanding this changes how behavior is interpreted. A person who struggles with motivation is not necessarily lazy. Their tonic dopamine may be low due to genetics, sleep deprivation, depression, or chronic stress. A person who seeks constant stimulation is not necessarily shallow. Their system may be calibrated for high novelty. A person who forms habits quickly is not weak-willed. Their learning circuits may be highly sensitive. These differences are biological. They are shaped by development, environment, and experience. They are not chosen. Dopamine itself is synthesized from the amino acid tyrosine. This process requires enzymes and occurs within neurons. Once produced, dopamine is stored in vesicles. When a neuron fires, these vesicles release dopamine into the synapse. Receptors on the next neuron bind it. Transporters then clear it away. Enzymes break it down. This cycle happens in milliseconds, billions of times a day. Every thought, every movement, every shift of attention relies on such exchanges. The mind is not separate from this. Thought is what this looks like from the inside. Seeing dopamine in this way strips it of mystique without stripping it of importance. It becomes neither villain nor hero. It becomes infrastructure. It is part of the machinery that allows a brain to orient in a changing world. The trouble arises when the environment overwhelms that machinery, when signals become constant, when unpredictability becomes engineered, when rest becomes rare. The system still does what it evolved to do. The outcome feels different. The goal of understanding dopamine is not to suppress it. It is to recognize the conditions under which it thrives and the conditions under which it becomes noisy. It is to see how attention is shaped. It is to understand why effort sometimes feels heavy and why novelty sometimes feels irresistible. This understanding becomes the foundation for everything that follows, because once dopamine is seen not as pleasure but as prediction, not as reward but as relevance, the patterns of modern life begin to make sense in a different way. And that clarity creates room for a gentler relationship with the mind as it moves forward into how these signals shape motivation itself. Inside the skull, beneath layers of bone and tissue, the brain is not arranged like a single control center. It is a network of specialized regions, each handling different aspects of perception, memory, movement, emotion, and planning. What people often imagine as the reward system is not one place. It is a distributed map of pathways that connect these regions in patterns shaped by evolution and experience. Dopamine travels along these pathways, linking sensation to action, prediction to movement, and memory to motivation. At the center of this map are a few small clusters of neurons deep in the midbrain. The ventral tegmental area and the substantia nigra contain most of the brain's dopamine-producing cells. These regions are tiny in physical size, yet they influence enormous territories. Their neurons send long axons outward, branching through the brain like roots spreading through soil. Along these paths, dopamine is released in different contexts, serving different functions depending on where it arrives. One major destination is the nucleus accumbens, a structure buried in the forebrain. This region integrates signals about internal state, sensory input, and goals. It sits at a crossroads between emotion and action. When dopamine reaches the nucleus accumbens, it alters how strongly the brain responds to potential outcomes. It changes how compelling something feels. It does not decide what you want. It changes how intensely the brain leans toward it. Another destination is the prefrontal cortex, especially areas involved in planning, working memory, and decision-making. Dopamine here influences focus, flexibility, and the ability to hold goals in mind. Too little dopamine in these regions can make thought feel foggy and effortful. Too much can make thinking scattered or rigid. The balance affects how easily attention stays with the task and how well the mind shifts when conditions change. The amygdala, involved in emotional learning, also receives dopamine input. This shapes how strongly experiences are tagged with emotional significance. It influences which memories carry weight and which fade. Events associated with unexpected outcomes are more likely to be remembered. Dopamine helps stamp those moments. The hippocampus, central to memory formation, interacts with dopamine signals as well. When something novel occurs, dopamine release increases the likelihood that the experience will be encoded. This is why first experiences tend to stand out. Novelty itself is a trigger. The brain treats the unfamiliar as potentially important. The basal ganglia, a collection of structures involved in movement and habit, are saturated with dopamine receptors. These circuits determine which actions are selected and which are suppressed. Every moment, multiple potential actions are possible. Move, stay still, speak, remain silent, reach, withdraw. Dopamine biases this competition. It makes some actions easier to initiate than others. Taken together, these pathways form a map that links perception, memory, emotion, and movement. It is not a single reward center. It is a network that continuously evaluates what is happening and adjusts the brain's orientation toward the future. When something unexpected occurs, dopamine neurons change their firing. This change propagates through the network. It alters how the brain updates its model of the world. If a cue reliably predicts a positive outcome, that cue gains power. It becomes noticeable. It stands out. If an outcome fails to arrive, the cue loses strength. Over time, this process builds an internal landscape of meaning. Some stimuli become charged, others fade. The map is personal. It reflects individual history. Two people can experience the same environment and develop very different internal priorities because their dopamine systems have learned different patterns. A child praised for drawing may develop strong dopamine responses to creative tasks. Another child praised for athletic performance may develop a different map. A person who finds safety in routines may have circuits that respond strongly to predictability. Another who grew up in uncertainty may be tuned toward change. This map is not fixed. It is plastic. It changes with experience. It is reshaped by stress, sleep, illness, medication, and environment. It is influenced by genetics, but not determined by them. Importantly, the map does not track what is good in an abstract sense. It tracks what has mattered before. It tracks patterns of outcome. It reflects reinforcement, not wisdom. If something reliably produces a noticeable effect, the system learns it. That effect might be nourishment, relief, stimulation, or escape. The system does not distinguish between growth and avoidance. It distinguishes between change and no change. This is why habits can form around behaviors that do not improve life. The brain learns what reduces discomfort or increases stimulation in the short term. Dopamine marks those patterns. The map adjusts. The next time a similar state arises, the pathway activates. The brain's reward map is therefore less about pleasure and more about direction. It points the organism toward what has previously altered internal state. It does not care whether that alteration is healthy. In a stable environment, this works well. Food reduces hunger. Social connection reduces isolation. Skill practice leads to improvement. The map aligns with a long-term benefit. In a highly engineered environment, the alignment can break. Stimuli can be designed to trigger the system without providing nourishment. The map still updates. The circuits still learn. The brain still leans. The map is also hierarchical. Some pathways respond to primary needs such as food, safety, and warmth. Others respond to secondary goals such as status, achievement, and novelty. The prefrontal cortex can modulate these signals, delaying action, weighing consequences, but that modulation itself depends on dopamine. The ability to hold a long-term goal in mind is supported by the same chemical that drives immediate pursuit. This creates tension. The same system that allows planning also amplifies temptation. The same circuits that support focus can be hijacked by distraction. The map does not contain a separate layer for virtue. It contains gradients of salience. When people speak of willpower, they often imagine a force that stands apart from chemistry. In reality, self-control is implemented by circuits that require neurotransmitters to function. The ability to resist a pull depends on the strength of competing signals. Those signals are also shaped by dopamine. This does not mean resistance is impossible. It means it is embodied. It is not a pure act of mind. It is a negotiation between circuits. Understanding the brain's reward map reframes behavior. Instead of asking why a person repeatedly chooses something, it asks what the map has learned. Instead of attributing failure to character, it examines history. A person who repeatedly checks a phone is not necessarily seeking pleasure. They may be responding to a map trained by intermittent reinforcement. A person who struggles to begin tasks may have a map shaped by fatigue, stress, or depression. The terrain they are moving through is different. This map also explains why change is difficult. When a new behavior is attempted, it has little representation. The pathways are weak. Dopamine does not rise strongly in response to its cues. The action feels heavy. The old pathways remain strong. Their cues still trigger expectation. The brain leans toward what it knows. Change requires repeated experiences that generate new prediction errors. It requires the system to learn that a different pattern produces a meaningful outcome. This is slow. It is biological. It is not solved by insight alone. Sleep plays a role here. During rest, the brain replays patterns. Synapses are adjusted. The map is updated. Chronic sleep deprivation impairs this process. Learning becomes noisy. Habits become harder to reshape. Dopamine signaling itself is altered by lack of rest. Stress also reshapes the map. Under chronic stress, the brain prioritizes immediate relief. Dopamine responses shift. Long-term goals lose salience. Short-term escape gains weight. This is not weakness. It is an adaptive response to threat. In an unsafe environment, the future is uncertain. The system favors now. The reward map is therefore dynamic. It reflects both past and present conditions. It is a living structure. Seeing it as a map rather than a tyrant changes the relationship with it. A map can be read. It can be understood. It can be redrawn over time. It is not an enemy. It is a record of learning. Dopamine does not force action. It marks possibilities. It highlights paths. The brain still moves along them using many systems at once. Thought, memory, emotion, and context all participate. This network is what allows learning to be efficient. It is what lets organisms adapt. It is what makes curiosity possible. Without it, behavior would be random. With it, experience leaves traces. The same system that can trap also enables growth. The same circuits that repeat can also discover. The map does not only point backward, it can be updated. Understanding this map sets the stage for exploring how anticipation becomes more powerful than arrival, how wanting often outweighs having, and why the future pulls more strongly than the present. That pattern emerges directly from the way these circuits handle prediction. There is a familiar pattern in human experience that feels almost universal. The build-up to something often feels more vivid than the moment it finally arrives. The days before a trip can feel charged with energy, while the first evening at the destination may feel strangely flat. The idea of starting a new project can feel exciting, yet the first hour of actually doing it can feel heavy. Even small things follow this rule. Waiting for a message can feel more intense than reading it. Planning a meal can feel more satisfying than eating it. This is not a failure of imagination or gratitude. It is a reflection of how the brain's motivational systems are wired. Dopamine rises most strongly in anticipation, not in fulfillment. It is the chemical signature of what might be, not what is. The system evolved to energize pursuit, not to prolong arrival. From the brain's perspective, a goal that has been achieved no longer needs energy. Once food is eaten, movement can stop. Once shelter is reached, vigilance can relax. The function of dopamine is to bridge the gap between current state and potential improvement. It creates momentum. When the gap closes, the signal fades. This is why wanting often feels sharper than having. During anticipation, the brain is actively predicting a change. Dopamine neurons increase their firing as cues accumulate. Each sign that the reward is approaching strengthens the signal. The system is engaged. Attention narrows. Energy mobilizes. When the reward finally arrives, the prediction is resolved. The brain registers that what was expected has occurred. The dopamine signal does not need to spike, because there is no longer a discrepancy between expectation and reality. The system has done its job. This does not mean enjoyment disappears. Other neurotransmitters and systems contribute to pleasure and satisfaction. Endorphins, opioids, and other circuits create the feeling of contentment. But those systems are quieter. They do not create urgency. They do not pull the mind forward. They settle rather than drive. In a world oriented toward constant motion, the settling phase can feel underwhelming. The brain is tuned to notice change. When stimulation drops, attention wanders. The absence of anticipation can feel like emptiness. This pattern shapes many behaviors. It is why shopping can feel more exciting than owning. It is why browsing can feel more engaging than choosing. It is why planning can feel better than doing. The system rewards the chase more than the capture. In environments where goals were concrete and necessary, this worked well. Anticipation pushed organisms toward food, shelter, and safety. Once those were secured, rest followed. The cycle had a rhythm. In modern environments, the chase rarely ends. There is always another item, another message, another update. Anticipation becomes continuous. The system is rarely allowed to settle. Dopamine rises again and again without the natural pause that once followed completion. This can create a subtle dissatisfaction. The brain remains oriented toward what comes next. The present moment feels like a staging area rather than a destination. Attention leans forward. This does not mean people are incapable of contentment. It means the system that evolved to motivate action is being continuously engaged by signals that never fully resolve. The gap between expectation and fulfillment becomes a permanent feature. The same mechanism explains why habits form around checking. Each check holds the possibility of something new. Most checks yield nothing of interest, but occasionally there is a reward. That unpredictability drives learning. Dopamine spikes when the outcome is better than expected. The brain strengthens the connection between the cue and the behavior. Over time, the anticipation itself becomes the draw. The hand moves before the thought forms. The system has learned that this pattern sometimes produces a signal. The cost of missing that signal feels larger than the cost of checking. This is not compulsion in a dramatic sense. It is the normal operation of a learning system exposed to intermittent reinforcement. Anticipation also shapes emotion. Anxiety is, in part, a form of negative anticipation. The brain predicts a potential threat. Dopamine interacts with stress systems to keep attention oriented toward what might go wrong. The mind rehearses futures that have not occurred. The same circuitry that imagines reward can imagine danger. In both cases, the present becomes secondary. The body is here, the mind is elsewhere. Understanding this pattern helps explain why mindfulness practices often emphasize returning to the present. They are not mystical techniques. They are ways of disengaging the anticipatory loop. When attention is placed on immediate sensation, prediction quiets. Dopamine-driven forward lean softens. This does not remove motivation. It temporarily suspends the chase. It allows the nervous system to register that nothing needs to be pursued in this moment. In everyday life, moments of contentment often occur when anticipation is low. Sitting after a meal, lying down at the end of a long day, watching something familiar. These states involve a shift from pursuit to presence. Dopamine activity decreases. Other systems dominate. In a culture that equates value with progress, these states can feel unproductive. But biologically, they are necessary. They allow recovery. They allow integration. They prevent the system from remaining in a constant state of drive. The tension between wanting and having is therefore not a flaw. It is an artifact of a system designed for movement. The problem arises when movement has no natural end point. Understanding that wanting feels better than having is not a call to abandon goals. It is an invitation to recognize that the brain is built to favor the slope over the plateau. It helps explain why achievement often feels anticlimactic, and why the next goal appears so quickly. This pattern becomes especially visible in long-term pursuits. A student works toward a degree for years. The day it is awarded feels brief. The system that carried them forward now has no target. The map must be redrawn. A new gap must be found. The same occurs in careers, relationships, and personal projects. The brain is more engaged by becoming than by being. This does not mean fulfillment is impossible. It means fulfillment is not driven by the same circuitry as pursuit. It arises from different systems, quieter ones that are easily drowned out by constant anticipation. Learning to notice this distinction changes how experiences are interpreted. The fading of excitement after arrival is not ingratitude. It is chemistry doing what it evolved to do. The challenge of modern life is not that anticipation exists. It is that anticipation is continuously triggered without resolution. The system is kept in a state of forward lean. The present becomes thin. As this becomes clearer, it sets the stage for understanding how dopamine shapes movement itself, how it turns intention into action, and why effort can feel either light or impossibly heavy, depending on the state of these circuits. Movement is often treated as a matter of will. We speak about getting ourselves to act, pushing through resistance, forcing momentum into existence. From the outside, motion appears simple. A body stands, walks, reaches, types. From the inside, movement begins long before muscles contract. It begins in circuits that evaluate possibility, effort, and outcome. It begins in the subtle chemistry that determines whether an intention becomes an action or remains only a thought. Dopamine is central to this translation. It does not move muscles directly. It changes the conditions under which movement becomes likely. It influences how heavy or light an action feels before it happens. It alters the threshold between thinking and doing. In the motor system, dopamine shapes how easily signals pass through the basal ganglia, a collection of structures that sit between intention and execution. Every moment the brain generates multiple potential actions. Some are external, like standing or speaking. Others are internal, like shifting attention or recalling a memory. These possibilities compete. The basal ganglia help decide which one wins. Dopamine modulates this competition. When dopamine is present at healthy levels, the selected action flows smoothly. When it is low, initiation becomes difficult. The person may know what they want to do. The intention exists. The body does not follow easily. This is visible in Parkinson's disease. People with this condition often describe feeling stuck. They can think about moving. They can plan. Yet starting a movement can feel like pushing against invisible resistance. This is not a lack of desire. It is a lack of chemical support in the pathways that enable intention to become motion. A similar, though less extreme pattern appears in everyday life. On days when energy feels low, tasks feel heavier before they begin. The mind may know what needs to be done, but the body does not respond. The gap between intention and action widens. Dopamine participates in closing that gap. In motivational circuits, it alters how effort is evaluated. The brain is constantly estimating cost. Every action has a metabolic price. Moving requires energy. Thinking requires energy. The system balances expected benefit against expected effort. When dopamine rises in response to a potential outcome, it tilts that balance. The benefit feels closer. The effort feels more acceptable. The brain becomes more willing to invest. This is why motivation feels like energy. It is not merely desire. It is the sensation that action is possible. It is the lowering of internal friction. When dopamine is low, the same action feels more costly. The hill becomes steeper. This does not mean the person lacks discipline, it means the internal gradient has changed. Depression illustrates this well. One of its core features is anhedonia, the reduced ability to feel pleasure, but another, often more disabling feature is psychomotor slowing. Movements feel heavy, decisions feel laborious, starting anything feels exhausting. This is not simply sadness, it reflects altered dopamine signaling. The circuits that normally assign weight to potential outcomes do not respond as strongly. The brain predicts less reward from action. Effort feels unjustified. The person may still care, they may still want improvement, but the chemical support for initiation is impaired. The same pattern appears under chronic stress. Prolonged activation of stress hormones alters dopamine function. The brain shifts toward immediate survival. Long-term goals lose salience. Actions that once felt meaningful now feel distant. The system conserves energy. Sleep deprivation has a similar effect. Dopamine receptors become less responsive. Baseline levels change. The world feels duller. Tasks feel heavier. The body resists motion. These states are often interpreted as laziness. In reality, they reflect altered internal conditions. The machinery that converts intention into movement is running under strain. On the other side of the spectrum, elevated dopamine can produce restlessness. Actions begin quickly. The mind jumps. Movement feels urgent. In extreme forms, this appears in mania, where activity becomes excessive and unregulated. The system overestimates reward and underestimates cost. Healthy motivation exists in a middle range. It allows action without compulsion. It supports effort without frenzy. It gives energy without erasing caution. Dopamine contributes to this balance by modulating both readiness and restraint. In the basal ganglia, it affects not only the pathways that promote movement, but also those that inhibit competing actions. It sharpens selection. It reduces noise. This means motivation is not just about starting, it is also about choosing. The brain must decide not only to act, but how. In everyday terms, this is felt as clarity. On days when motivation is high, priorities feel obvious. The mind settles on a task. Movement follows. On days when dopamine signaling is impaired, everything feels equally heavy. Choice itself becomes exhausting. This is why lists and plans sometimes fail. They provide information, not energy. The brain may know what to do. The chemistry may not support doing it. External structure can help. Deadlines, social accountability, and routines act as surrogate signals. They create predictability. They generate anticipation. They can provoke dopamine release even when internal drive is low. This is not weakness. It is alignment. The brain evolved in social contexts where external cues mattered. Movement was often coordinated with others. Structure provided meaning. In modern life, many tasks are abstract. Their outcomes are distant. The dopamine system responds weakly to distant reward. It evolved for immediate consequences. This mismatch explains why starting long-term projects feels harder than responding to urgent messages. The latter provide immediate feedback. The former require sustained effort with delayed outcome. Understanding this reframes productivity. It becomes less about forcing will and more about shaping conditions. It becomes about aligning tasks with how the brain assigns value. Breaking a task into smaller steps increases the frequency of feedback. Each completed step creates a small prediction error. Dopamine rises. The map updates. The next step feels lighter. This is not a trick. It is how learning works. The brain moves more easily when it receives signals along the way. Movement is therefore not purely mechanical. It is guided by expectation. It is influenced by memory. It is shaped by internal state. Even attention is a form of movement. Shifting focus from one thought to another requires neural energy. Dopamine modulates this as well. It affects cognitive flexibility. It influences how easily the mind lets go. When dopamine is balanced, attention can move smoothly. When it is low, the mind becomes sticky, thoughts repeat, rumination sets in. When it is high, attention may jump too quickly. These patterns are often mistaken for personality traits. They are in part chemical states. This does not mean people are reducible to molecules, it means experience has a physical substrate. The feeling of being able to begin is not abstract, it is embodied. Motivation, then, is not a single force. It is the emergent result of circuits evaluating cost and benefit under specific conditions. Dopamine is one of the key modulators of that evaluation. When it functions well, action feels possible. When it falters, action feels heavy. When it overfires, action becomes scattered. Seeing this allows a gentler interpretation of inertia. It is not always resistance. Sometimes it is a signal that the system lacks support. This perspective opens the door to understanding how repeated actions become automatic, how effort fades with practice, and how the brain gradually replaces conscious choice with habit. Habits are often spoken about as if they are shortcuts the mind takes, ways of conserving effort by turning deliberate actions into automatic routines. That description is partly true, but it hides the deeper biological machinery that makes habits possible. A habit is not simply a repeated behavior. It is a pattern encoded in neural circuits, shaped by chemistry, reinforced by experience, and maintained by prediction. Dopamine sits at the center of this process, not as a reward in itself, but as the signal that teaches the brain which patterns are worth repeating. Every habit begins as a choice. The first time you perform an action, it requires attention. You think about it. You decide. You guide your body through unfamiliar steps. The brain is actively involved in planning and monitoring. During this early phase, dopamine responds strongly to outcomes. If the result is better than expected, a prediction error occurs. Dopamine rises, synapses strengthen. The brain marks the sequence as meaningful. With repetition, the brain begins to compress the pattern. Control shifts from regions involved in conscious planning toward deeper structures in the basal ganglia. The behavior becomes faster. It requires less thought. Eventually, the cue alone is enough to trigger the sequence. The mind no longer needs to decide. The body begins before awareness catches up. This transition is not a metaphor. It reflects a physical relocation of control. Early learning engages the prefrontal cortex where goals are held in mind. As the pattern stabilizes, activity moves toward the dorsal striatum, a region specialized for routine. Dopamine supports this shift. It reinforces the connections that link cue to action to outcome. Once a habit is established, dopamine no longer spikes at the reward, it spikes at the cue. The brain has learned that the cue predicts a change. The anticipation itself becomes the driver. The action follows automatically. This is efficient. It frees cognitive resources. It allows complex behavior to unfold without constant supervision. Walking, typing, brushing teeth, driving familiar routes, all rely on this mechanism. Without habits, daily life would be exhausting. The same mechanism applies to less helpful behaviors. The brain does not distinguish between brushing teeth and checking a phone. It distinguishes between patterns that reliably produce a noticeable outcome and those that do not. If checking produces stimulation, relief, or distraction, dopamine reinforces it. Over time, the behavior becomes decoupled from conscious intent. You may not feel a strong desire. You may even feel resistance. Yet the hand moves. The sequence begins. The habit has become a reflex. This does not mean the person lacks control. It means the control has been distributed into deeper circuits. To change the behavior, those circuits must be retrained. Insight alone is not enough. The map must be updated through new experiences. Breaking a habit therefore feels different from forming one. Formation often feels light. The early phase is marked by novelty. Dopamine responds strongly. The brain is engaged. Change feels interesting. Breaking a habit feels heavy. The old cue still triggers expectation. Dopamine still rises. The body still prepares. Resisting requires the prefrontal cortex to intervene. That intervention itself consumes energy. It feels like effort. Each time the habit is not completed, a small prediction error occurs. The expected outcome does not arrive. Dopamine dips. Over time, the association weakens. But this process is slow. The brain is conservative. It does not abandon a pattern easily, especially one that has worked reliably in the past. This is why habits persist even when they no longer serve. The system is not evaluating long-term meaning. It is responding to historical reliability. Stress complicates this. Under stress, the brain shifts control toward habitual circuits. The prefrontal cortex becomes less effective. The system favors speed over flexibility. In uncertain environments, this is adaptive. It allows rapid response. In modern life, stress is often psychological rather than physical. The same shift occurs. Under pressure, people fall back on habits. They reach for what is familiar. This is not regression, it is biology. Sleep also plays a role. During rest, the brain consolidates patterns. Synaptic changes are stabilized. Habits become more ingrained. Sleep deprivation disrupts this process. Learning becomes erratic. Old habits persist. New ones struggle to form. This means that change is not only about effort during the day, it is about conditions at night. The brain rewrites itself in cycles. Dopamine interacts with these cycles. Without rest, the system becomes noisy. Understanding this changes how habit is perceived. It is not a moral failing. It is a learning artifact. It is the brain doing what it evolved to do. This perspective removes some of the drama from behavior change. It becomes a matter of training rather than judgment. The goal is not to overpower the system. It is to give it new data. Small changes matter because they produce frequent prediction errors. Each time a cue appears and the old behavior does not follow, the brain updates. Each time a new behavior produces noticeable outcome, the new pathway strengthens. This is why environment matters. Removing cues reduces the frequency of activation. Adding friction slows automaticity. Creating new cues supports alternative patterns. These are not tricks. They are ways of shaping the learning landscape. Dopamine does not lock habits in place. It keeps them alive as long as they remain predictive. When conditions change consistently, the map changes. The difficulty lies in the time scale. The brain learned the habit over many repetitions. It will unlearn it over many mismatches. Patience is not a virtue here. It is a requirement imposed by biology. The same system that makes habits stubborn also makes skill acquisition possible. Practice is habit formation in a constructive direction. Each repetition strengthens pathways. Each success generates a small prediction error. The brain learns. Musicians, athletes, and craftspeople rely on this. What begins as awkward becomes fluid. Effort fades. Movement becomes automatic. Dopamine reinforces the pattern. This is the positive face of the same mechanism. Habit is not inherently limiting. It is a tool. It can imprison or liberate depending on what it encodes. Seeing habits through this lens dissolves some of the shame around them. It reveals behavior as the product of training, not character. It shows that the brain is loyal to its own history. Change, then, is not about declaring a new identity. It is about giving the system repeated experiences that contradict its predictions. It is about allowing dopamine to teach something new. This process is quiet. It unfolds beneath awareness. It does not respond to slogans. It responds to patterns. And because it is slow, it benefits from conditions that reduce noise, lower stress, better sleep, simpler environments. These do not guarantee change. They make learning possible. Dopamine is not the enemy of habit. It is the reason habits exist at all. It is the system that allows experience to shape behavior. Understanding this opens the way to examining how pleasure, addiction, and the modern environment interact with the same circuitry, often overwhelming it with signals it was never designed to handle. pleasure is one of the most misunderstood experiences in the brain. It is often treated as the goal of dopamine, the destination that the system exists to deliver. In reality, pleasure and dopamine are related but distinct. Pleasure is a felt experience that arises from several interacting systems. Dopamine is not the feeling itself. It is part of the machinery that leads towards situations where pleasure might occur. The confusion between these two has shaped how people think about motivation, addiction, and modern life. When something feels pleasant, other neurotransmitters are often more directly involved. Endogenous opioids, for example, contribute to feelings of comfort and satisfaction. Endocannabinoids influence relaxation and ease. Oxytocin plays a role in social warmth and bonding. These systems are associated with states of contentment, connection, and relief. Dopamine, by contrast, is most active before pleasure. It rises in response to cues that predict something that might change your internal state. It prepares the brain. It creates a sense of pull. It sharpens attention. It mobilizes energy. The pleasure that follows is not caused by dopamine in the simple sense. It is enabled by the behavior dopamine helps initiate. In natural environments, this separation worked well. Hunger produced a drive to seek food. Dopamine supported that search. Eating produced satisfaction. Afterward, the system settled. There was a clear arc from anticipation to fulfillment to rest. In the modern world, that arc is often interrupted. Many experiences provide anticipation without meaningful resolution. Others provide stimulation without nourishment. The brain receives repeated cues that promise change, but the resulting pleasure is brief or shallow. The system is activated again almost immediately. This pattern is central to understanding addiction. Addiction is not simply excessive enjoyment. It is a disorder of learning and prediction. Substances and behaviors that strongly activate dopamine pathways teach the brain that a particular cue reliably produces a powerful internal shift. The prediction becomes intense, the cue becomes charged, the behavior becomes prioritized. Drugs that directly increase dopamine release bypass the normal teaching process. They produce large, unearned signals. The brain interprets these as evidence that something extraordinarily important has occurred. Synapses change rapidly. The map is rewritten. Natural rewards rarely produce dopamine surges of that magnitude. They are constrained by biology. Food, sex, social contact, and achievement all produce signals within a certain range. Psychoactive substances can exceed that range. They hijack the learning system. This does not mean addiction is a moral collapse. It is a predictable response of a learning system exposed to extreme signals. The brain does exactly what it evolved to do. It prioritizes what appears to matter most. Over time, the system adapts. Receptors become less sensitive. Baseline dopamine function shifts. The person experiences less pleasure from ordinary activities. The world feels flat. The substance or behavior becomes necessary not to feel good, but to feel normal. This is not because the person lacks gratitude. It is because the calibration of the system has changed. The modern world contains many experiences that mimic aspects of this pattern without involving substances. Variable reward schedules, endless novelty, and constant availability of stimulation engage the same circuits. The signals are smaller than those produced by drugs, but they are frequent and unpredictable. Each notification holds the possibility of social connection. Each scroll holds the possibility of interest. Each click holds the possibility of surprise. Most of the time, nothing meaningful happens. Occasionally something does. The brain learns. This is not accidental. Many systems are designed around these principles because they are effective. They capture attention. They keep users engaged. They exploit the same learning mechanisms that once guided organisms toward survival-relevant information. The result is a nervous system that is rarely allowed to settle. Anticipation becomes continuous. The baseline shifts. The absence of stimulation feels like deprivation. This does not mean technology is inherently harmful. It means it interacts with ancient circuits. It means the cost of constant engagement is carried by systems that evolved for scarcity. In this environment, pleasure becomes fragmented. Satisfaction becomes brief. The mind moves quickly from one target to the next. The space between wanting and having collapses into a narrow loop. People often interpret this as personal dissatisfaction. They feel restless. They feel unfulfilled. They assume something is wrong with them. In reality, their nervous systems are responding normally to abnormal conditions. Addiction exists on a spectrum. At one end are substances that directly alter dopamine release. At the other are behaviors that provide intermittent reinforcement. In between lie patterns that can become compulsive without involving chemicals. The common thread is not pleasure, it is prediction. The system becomes oriented toward a cue. The mind anticipates, the body prepares, the behavior follows. Breaking these patterns requires more than insight. It requires altering the learning environment. It requires creating conditions where prediction errors occur in new directions. It requires time. This is why willpower alone is unreliable. Willpower is a function of prefrontal circuits that themselves depend on dopamine. Under fatigue or stress, those circuits weaken. The deeper habit systems dominate. Recovery, whether from substance addiction or from compulsive patterns, is therefore not about fighting chemistry. It is about retraining it. It is about reducing exposure to cues. It is about increasing alternative sources of meaningful signal. It is about restoring baseline function through sleep, nutrition, and stability. Pleasure itself is not the enemy. The brain is designed to enjoy. The problem arises when the systems that generate anticipation are continuously engaged without allowing the systems of satisfaction to operate fully. In a balanced cycle, effort leads to outcome. Outcome leads to rest. Rest restores capacity. In a disrupted cycle, anticipation leads to stimulation. Stimulation fades quickly. Anticipation resumes. This loop produces a sense of constant incompleteness. The mind is always reaching. The present feels insufficient. Understanding this pattern shifts the question from why am I not satisfied to what is my system being trained to expect. It reframes restlessness as a signal of mismatch rather than a personal defect. The goal is not to eliminate stimulation. It is to restore rhythm. It is to allow arcs of pursuit and completion to exist again. It is to create space where satisfaction can last long enough to register. This requires conditions that modern environments rarely provide by default. It requires boundaries. It requires pauses. It requires periods where nothing new arrives. These conditions do not feel immediately rewarding. They do not produce spikes. They feel quiet. They allow other systems to come online. The challenge is that a brain accustomed to frequent dopamine signals interprets quiet as absence. It feels like something is missing. That feeling is not a verdict. It is a symptom of recalibration. Over time, as the baseline adjusts, quiet becomes neutral again. Contentment becomes possible without constant input. The world regains texture. This is not about rejecting modern life. It is about understanding how deeply it speaks to ancient circuitry. It is about recognizing that pleasure and addiction are not opposites. They are points on the same learning curve. The same system that allows joy also allows compulsion. The difference lies in magnitude, frequency, and context. Dopamine does not distinguish between nourishment and noise. It responds to change. It responds to unpredictability. It responds to cues that promise alteration. To live well with such a system is not to suppress it. It is to give it environments that align with its design. It is to allow anticipation to have endpoints. It is to allow satisfaction to unfold without being immediately displaced. This understanding naturally leads into the social dimension of dopamine, because many of the most powerful cues in modern life are not objects or substances, but signals of approval, belonging, and status. Human beings are social organisms long before they are rational ones. Long before written language, long before agriculture, long before cities or tools, survival depended on staying connected to others. Food was shared, danger was detected together. Children survived because someone noticed them. Exile meant vulnerability. Belonging meant protection. Over hundreds of thousands of years, nervous systems adapted to this reality. The brain learned to treat social signals as biologically significant. Dopamine became one of the chemicals that translated social information into motivation and behavior. Approval is not merely emotional, it is chemical. A smile, a nod, a word of praise, a sign of recognition can produce measurable changes in dopamine activity. These signals do not feel abstract, they feel energizing. They create warmth, alertness, and a subtle sense of orientation. The brain interprets them as markers of safety and opportunity. They say, in biological terms, you're aligned with the group. Rejection carries the opposite signal. Averted eyes, silence, criticism, or exclusion trigger stress circuits. Dopamine responses shift. The brain becomes vigilant. Attention turns inward. The system prepares for potential threat. This is not oversensitivity, it is inherited survival logic. Social dopamine does not exist in a single location. It emerges from the same reward map that governs other forms of learning. The difference lies in the type of cues. Faces, voices, names, symbols of status, and group markers become triggers. The brain learns which social actions lead to positive outcomes and which lead to discomfort. A child quickly learns which behaviors earn smiles and which bring disapproval. Dopamine spikes when a parent's face lights up. Prediction errors occur. The brain strengthens those patterns. Over time, identity forms around what produces social alignment. This process is not conscious. It unfolds before memory. It shapes posture, tone, and attention. It teaches the nervous system how to belong. Status is part of this system. In social species, hierarchy reduces conflict. It clarifies roles. The brain therefore treats rank as meaningful. Signals of rising status often produce dopamine responses. Signals of falling status can feel like threat. This does not mean people are shallow. It means the nervous system is calibrated for group life. The brain tracks relative position because it once predicted access to resources and safety. In modern life, these ancient systems encounter symbolic environments. Status is signaled by numbers, images, titles, and metrics. Approval arrives as notifications. Belonging appears as inclusion in digital spaces. The same circuits respond. A like is not just information, it is a cue. It predicts social alignment. Dopamine responds, the brain learns. The unpredictability of these signals amplifies their effect. Sometimes a post receives attention, sometimes it does not. The brain cannot predict the outcome. Each check holds possibility. The system stays engaged. This is the same mechanism that underlies gambling. Variable reward schedules produce strong learning. The brain remains alert because the next signal might matter. Social platforms did not invent this response. They align with it. They provide environments where ancient circuits operate at modern scale. The result is a nervous system that treats symbolic approval with the weight once reserved for face-to-face belonging. The emotional impact is real because the chemistry is real. This does not mean social connection is artificial. It means the medium has changed faster than the brain. Offline, social feedback is slower and richer. Tone, expression and context moderate meaning. Online, signals are compressed. A single number stands in for complex response. The brain fills in the rest. Dopamine does not distinguish between a nod from a neighbour and a metric on a screen. It responds to salience. It responds to learned cues. This can reshape behaviour. People may begin to act in ways that maximise signal rather than meaning. They may orient toward what produces a response rather than what feels true. not because they are vain, but because their learning systems are adapting. Belonging is a biological need. The brain will seek it where it can. The same circuits also govern avoidance. Fear of social disapproval can suppress action. The brain predicts negative outcomes. Dopamine signaling shifts. The cost of expression feels high. Silence feels safer. This is not weakness. It is risk assessment performed by ancient machinery in modern contexts. Understanding social dopamine reframes many patterns. The pull to check messages, the sting of being ignored, the warmth of recognition, the anxiety of exposure. These are not overreactions. They are the outputs of systems built for survival in groups. The problem arises when the scale and speed of modern feedback overwhelm calibration. The brain evolved for small groups. It now receives signals from hundreds or thousands of people. Each signal carries weight. The system does not know how to average. This can lead to hypervigilance. The mind scans constantly. It anticipates response. It replays interactions. Dopamine and stress systems intertwine. Rest becomes difficult. Offline, social cycles include repair. Misunderstandings are resolved. Feedback is contextual. Online, signals can be ambiguous. Absence can feel like rejection. The brain fills gaps with prediction. This environment can shift baseline expectations. The nervous system may come to expect frequent social input. In its absence, it feels deprived. The quiet that once felt neutral now feels empty. This is not because connection is bad. It is because the pace has changed. Belonging is not optional. The brain treats isolation as threat. Loneliness alters dopamine function. It reduces responsiveness to reward. It increases sensitivity to stress. The system prepares for a world without support. This has physical consequences. Immune function changes. Sleep is disrupted. Motivation declines. The organism conserves energy. Social dopamine therefore sits at the intersection of chemistry and culture. It shapes how people present themselves. It influences risk-taking. It alters tension. Understanding this does not require withdrawal from social life. It requires recognizing that approval is a powerful signal. It requires noticing how often the brain is pulled outward by anticipation of response. The goal is not to stop caring. It is to restore proportion. It is to remember that the nervous system interprets symbols as survival cues. It is to provide context where connection is slower, deeper, and less contingent on metrics. Face-to-face interaction engages additional systems. Touch, timing, and shared environment provide richness. The brain receives multiple channels of information. Signals are balanced. In such contexts, dopamine responses are embedded in broader patterns. Anticipation resolves into presence. Belonging is felt rather than counted. This balance is not always available. Modern life demands digital interaction, but awareness allows modulation. It allows choice of rhythm. The same system that makes approval powerful also makes curiosity powerful. The desire to know what others think is linked to the desire to explore. Both arise from prediction. Both involve dopamine. Social learning is one of the brain's most efficient forms of learning. Watching others reduces risk. The brain therefore treats social information as high value. This is why gossip spreads. It contains data about the group. It signals norms. It teaches what leads to acceptance or exclusion. Dopamine reinforces this learning. It marks social patterns as meaningful. It encourages attention. The cost is that attention can become externally driven. The internal compass weakens. The brain waits for signal. Restoring that compass does not require isolation. It requires cultivating sources of meaning that do not depend on immediate response. It requires activities where feedback is intrinsic. Movement, craft, reading, listening. These engage other systems. They allow dopamine to operate in quieter ways. Belonging then becomes layered rather than singular. It includes both social alignment and self-alignment. The brain receives signals from within as well as from others. This sets the stage for understanding how novelty and curiosity operate, because the same circuits that respond to social signals also respond to the unknown. Curiosity is often spoken about as a personality trait, something some people possess in abundance while others seem to lack. In reality, curiosity is a biological function. It is an expression of the same learning machinery that drives habit, motivation, and anticipation. It is the brain's way of seeking information in environments where knowledge improves survival. Dopamine plays a central role in this process, not by making exploration pleasurable in itself, but by assigning value to the unknown. From the perspective of the nervous system, novelty is informative. Something unfamiliar might signal opportunity or danger. Either way, it matters. Dopamine neurons respond to novelty because it represents a deviation from prediction. When the brain encounters something it cannot fully model, dopamine activity increases, attention sharpens, memory systems engage. The organism becomes oriented toward learning. This response appears early in life. Infants turn toward new sounds. They stare at unfamiliar faces. They reach for objects they have never touched. These behaviors are not taught. They emerge from circuitry tuned to change. The brain is built to sample the world. Curiosity is therefore not a luxury. It is a survival strategy. In environments where resources are scattered and threats unpredictable, exploration reduces uncertainty. The organism that learns more quickly gains advantage. Dopamine supports this by marking novelty as salient. It does not guarantee that the experience will be enjoyable. It guarantees that the brain will pay attention. It says, "This is different. Update your model." The pleasure that sometimes accompanies discovery is a secondary effect. It arises when new information integrates smoothly, when understanding expands. But the initial drive is not about pleasure. It is about reducing uncertainty. This is why curiosity often feels restless. It is not calm. It is an itch. It pulls attention outward. It seeks resolution. In natural environments, this drive leads to exploration of territory, tools, and social patterns. It produces learning that is embodied and contextual. The organism encounters novelty, extracts information, and incorporates it into behavior. In modern environments, novelty is abundant and disembodied. New information arrives without physical movement. The brain receives constant deviation from expectation. Headlines, images, and updates flood the system. Each one is small. Together they create a continuous stream of novelty. Dopamine responds as it always has. It marks difference. It sharpens attention. It prepares learning. The problem is not that the system is malfunctioning. The problem is that it's being engaged without resolution. In physical exploration, novelty leads to knowledge that stabilizes. A place becomes familiar. A tool becomes mastered. The drive subsides. In digital exploration, novelty often replaces itself. One unknown leads to another. The model never settles. The brain remains in a state of partial anticipation. It is always updating. It rarely completes. This alters how curiosity feels. It becomes fragmented. It loses depth. Instead of sustained inquiry, attention hops. The system is still learning, but it is learning shallowly. This does not mean the brain is broken. It means the environment is shaped differently. Curiosity is closely linked to dopamine's role in prediction error. When something slightly violates expectation, dopamine rises. When the violation is too large, stress may dominate. The brain seeks a balance. It wants novelty within reach. This is why puzzles are engaging. They present manageable uncertainty. The brain can imagine resolution. Each step produces feedback. Dopamine reinforces progress. When uncertainty is overwhelming, curiosity collapses into anxiety. The system no longer perceives learning as possible. It shifts toward threat. Healthy exploration exists in a middle zone. The unknown is neither trivial nor terrifying. It is accessible. In modern life, that zone is often bypassed. Information arrives without effort. The brain does not need to move. The feedback loop is compressed. Learning becomes consumption. The dopamine system does not distinguish between discovering a new plant and discovering a new image. It responds to novelty. The body remains still. The mind moves. Over time, this can weaken the association between curiosity and embodied action. The brain learns that exploration does not require movement. It becomes a mental activity. This shift has subtle consequences. Physical exploration involves risk, effort, and context. It engages multiple systems. Digital novelty is low cost. It requires little energy. The system adapts. Curiosity becomes less about understanding and more about scanning. The goal is not resolution. It is stimulation. This is not a moral failing. It is a learning outcome. Understanding this allows curiosity to be reclaimed. It allows the system to be given environments where novelty leads to integration rather than endless deferral. Reading deeply, building something, learning a skill, walking unfamiliar routes. These engage the same circuitry in different ways. They provide novelty that resolves into familiarity. They allow the brain to close loops. When loops close, dopamine subsides. The system rests. Satisfaction emerges. In contrast, open loops keep the system activated. The mind remains slightly unsatisfied. Attention continues to seek. Curiosity is therefore not only about seeking, it is about arriving. The modern environment often emphasizes seeking without arrival. The brain is trained to stay in pursuit. Recognizing this does not require rejecting information. It requires choosing forms of novelty that allow completion. Curiosity thrives when it is paired with agency, when the organism can act, test, and learn. Passive novelty does not satisfy in the same way. Dopamine evolved to support exploration in physical space. It now supports exploration in symbolic space. The difference lies in closure. When learning is embodied, it changes the map. The brain gains new predictive power. Uncertainty decreases. The drive subsides. When novelty is endless, prediction never stabilizes. The system remains alert. This pattern links back to anticipation. Wanting is maintained. Having is deferred. The brain's drive to explore is not wrong. It is ancient. It is adaptive. The challenge is to give it environments where exploration leads somewhere. This becomes especially important in understanding how stress and cortisol interact with dopamine, because stress can both suppress curiosity and distort it, narrowing the field of exploration and reshaping what feels possible. Stress is often spoken about as an emotional state, something that happens in the mind when life becomes overwhelming. In the body, stress is a physiological cascade. It begins with perception, but it unfolds through chemistry. The brain detects threat, uncertainty, or excessive demand and signals the adrenal system. Cortisol and related hormones are released, heart rate changes, energy is mobilized, attention narrows, the organism prepares to respond. This response evolved to protect. In short bursts, it is adaptive. It sharpens focus. It increases readiness. It prioritizes survival over comfort. In a dangerous environment, this can save a life. The body becomes efficient. Blood is redirected. Muscles are primed. Digestion slows. Non-essential processes are postponed. The mind becomes vigilant. Dopamine does not shut down during this state. It shifts. In the early phase of stress, moderate increases in cortisol can actually enhance dopamine signaling in certain circuits. This is why urgency can feel energizing. A looming deadline can spark productivity. A challenge can sharpen motivation. In these moments, stress and dopamine work together. The system interprets the situation as demanding action, and dopamine helps mobilize it. But this balance is narrow. It depends on the stress being brief and resolvable. When stress becomes chronic, when the system remains activated day after day without recovery, the relationship between cortisol and dopamine changes. Receptors become less sensitive. Baseline dopamine levels shift. The brain adapts to a world it perceives as persistently unstable. From the nervous system's perspective, prolonged stress signals an environment where effort may not reliably lead to safety or reward. In such conditions, long-term investment becomes risky. The system begins to conserve. Motivation declines. Curiosity fades. Actions that once felt meaningful lose their pull. This is not a psychological attitude. It is a biological adjustment. Under chronic stress, dopamine signaling becomes blunted in circuits involved in anticipation and motivation. The same activities that once produced interest now feel flat. The world loses contrast. This state is often described as burnout or emotional exhaustion. People report feeling empty, detached, or unable to care. They still know what matters. They still hold values. But the chemistry that once translated those values into energy is impaired. At the same time, other pathways become more prominent. The brain shifts toward behaviors that provide immediate relief. Quick comforts gain weight. Distraction becomes appealing. Familiar habits become harder to resist. These patterns are not signs of weakness. They are the nervous system prioritizing short-term stabilization. Cortisol also affects the prefrontal cortex, the region involved in planning, impulse control, and perspective. Under sustained stress, prefrontal function weakens. The brain becomes less flexible. It relies more heavily on habitual circuits in the basal ganglia. Behavior becomes more automatic. This shift made sense in ancestral environments. Under threat, there is no time for deliberation. The organism falls back on what is known. In modern contexts, this can feel like being stuck. The person may notice themselves repeating patterns they would otherwise change. Sleep disruption compounds this process. Cortisol and melatonin interact. Chronic stress interferes with circadian rhythms. Falling asleep becomes harder. Sleep becomes lighter. The brain does not fully cycle through restorative phases. Dopamine systems fail to recalibrate. During healthy sleep, synaptic connections are adjusted. Noise is reduced. Learning is consolidated. Dopamine receptors regain sensitivity. Without this process, the system becomes increasingly rigid. Old patterns persist. New ones struggle to form. This is why chronic stress and sleep deprivation often travel together with reduced motivation and increased compulsive behavior. The nervous system is trapped in a defensive posture. The reward map changes under these conditions. The system learns that effort is costly and uncertain. It learns that the environment is unpredictable. It favors caution over exploration. It narrows the field of what feels possible. This narrowing is often experienced as loss of self. People say they no longer recognize who they are. They may feel disconnected from interests that once defined them. In reality, the map has been reshaped by chemistry. Stress also alters how prediction errors are processed. Under high cortisol, the brain becomes less responsive to subtle positive signals. Small wins fail to register. Dopamine spikes are muted. The system does not update easily. It becomes difficult to rebuild momentum. Negative signals, however, are amplified. The brain remains vigilant for threat. Errors loom larger than successes. This bias reinforces withdrawal. This is not pessimism, it is threat learning. Recovery from chronic stress therefore requires more than removing the stressor. It requires retraining the nervous system to trust that effort can lead to stable outcome. Dopamine circuits must be given repeated experiences of reliability. This cannot be rushed. The brain updates slowly. It needs consistent evidence. Small routines matter. Predictable rhythms matter. Activities where action leads to visible result matter. Walking, cleaning, cooking, writing a paragraph, completing a simple task. These provide closed loops. The system experiences completion. Each completion creates a small prediction error. Something expected to be difficult turns out to be manageable. Dopamine responds, the map adjusts. This is why recovery often begins with modest steps, not because ambition is wrong, but because the system cannot yet bridge large gaps. It must relearn that movement is safe. Stress does not simply reduce energy, it reshapes what feels worth energy. Understanding this reframes many struggles. Difficulty initiating does not mean lack of desire. It means the bridge between intention and action has weakened. That bridge is chemical. The fragility of dopamine's balance lies in its dependence on context. It functions best in environments that provide a sense of stability. It falters in environments that signal constant threat. Modern life often combines high demand with low resolution. Tasks accumulate. Messages never end. Work spills into rest. The system receives continuous signals of unfinished business. The brain interprets this as unresolved threat. Cortisol remains elevated. Dopamine adapts. This does not require dramatic trauma. It can emerge from sustained pressure. Rest is not a luxury in this context. It is a biological necessity for recalibration. Without rest, the system cannot return to baseline. Rest does not only mean sleep, it means periods where no action is required, where nothing is expected, where the nervous system can downshift. These periods allow cortisol to fall, they allow dopamine receptors to recover, they allow the reward map to soften. Many people resist rest because it feels unproductive. In a stressed brain, stillness can feel unsafe. The system has learned that demand is constant. Silence feels like neglect. This is not laziness, it is adaptation. Reintroducing rest therefore requires patience. The system may initially respond with unease. That unease is not a signal that rest is wrong. It is a signal that the system has been trained to remain alert. Over time, as cortisol decreases and dopamine sensitivity returns, rest begins to feel neutral again, then restorative. This is not self-care as a slogan, it is neurobiology. Stress also interacts with meaning. When the brain perceives that effort lacks purpose, dopamine engagement drops. The system conserves. This is why environments that feel arbitrary or misaligned drain motivation more quickly than those that feel coherent. Meaning is not abstract to the brain, it is encoded in prediction. When actions reliably connect to outcomes that matter, dopamine supports engagement. When that link breaks, energy fades. This is why chronic misalignment is exhausting. The system is asked to invest without return. Restoring balance therefore involves not only reducing stress, but restoring coherence. The brain needs to experience that actions make sense. This may involve changing environments, it may involve redefining scope, it may involve simplifying. The nervous system is not impressed by ambition. It responds to reliability. Dopamine's fragility lies in its role as a bridge. It connects present effort to future outcome. Stress erodes trust in that bridge. Rebuilding it is a gradual process. It unfolds through repeated, modest experiences of success and safety. This sets the stage for understanding how sleep and circadian rhythms interact with dopamine, because sleep is the primary mechanism through which the brain restores that bridge and re-establishes the chemistry of possibility. Sleep is often treated as the absence of wakefulness, a passive state where nothing happens. In biological terms, sleep is one of the most active and organized processes the brain performs. It is not downtime, it is maintenance, it is calibration. It is the period in which the nervous system restores balance, reorganizes information, and resets the chemical conditions that make waking life possible. Dopamine is deeply entangled with this process. Its signaling patterns change across the day. Its receptors fluctuate in sensitivity. Its interactions with other neurotransmitters depend on whether the brain is aligned with its internal clock. When sleep is disrupted, dopamine does not simply continue unchanged. It adapts, often in ways that make waking life feel distorted. Every cell in your body contains molecular clocks. These clocks regulate gene expression on a roughly 24-hour cycle. In the brain, a small cluster of neurons in the hypothalamus acts as the master coordinator. This region synchronizes internal rhythms with external light. When light enters the eyes, signals travel directly to this clock. Hormone release shifts, body temperature changes, the brain prepares for wakefulness. Dopamine follows this rhythm. Baseline levels vary across the day. Sensitivity of receptors changes. In the morning, the system is primed for engagement. In the evening, it gradually downshifts. This is not arbitrary. It reflects millions of years of adaptation to a rotating planet. When the circadian system is aligned, motivation, attention, and mood fluctuate predictably. Energy rises after waking. It peaks in the afternoon. It declines in the evening. The brain expects rest. Dopamine signaling becomes quieter. Other systems take precedence. Artificial light, irregular schedules, and late stimulation disrupt this pattern. The brain receives conflicting signals. Melatonin release is delayed. Cortisol remains elevated. Dopamine rhythms flatten. This flattening has consequences. When dopamine does not cycle, the contrast between effort and rest blurs. Motivation feels erratic. Tasks feel heavier in the morning. Alertness feels artificial in the evening. Sleep deprivation amplifies these effects. Even one night of insufficient sleep alters dopamine receptor availability. The brain compensates by increasing dopamine release in some regions. This can produce a temporary sense of alertness. It feels like being wired. But this compensation is inefficient. It depletes resources. It masks fatigue rather than resolving it. Over time, chronic sleep loss reduces receptor sensitivity. The system becomes noisy. The same amount of dopamine produces less effect. The world feels dull. Motivation drops. The brain seeks stronger stimulation to achieve the same impact. This pattern mirrors addiction in miniature. The baseline shifts. Ordinary experiences feel flat. The system reaches for intensity. Sleep is the primary mechanism by which this drift is corrected. During deep sleep, dopamine neurons change their firing patterns. Receptors are resensitized. Synaptic noise is reduced. The reward map is recalibrated. Memory consolidation during sleep also interacts with dopamine. Experiences tagged as significant during the day are replayed. Circuits strengthen. Learning stabilizes. Without sleep, this replay is incomplete. The brain remains in a provisional state. This is why sleep deprivation impairs learning. It is not only that attention is reduced, it is that the chemical environment required for consolidation is missing. Circadian misalignment produces similar effects even when total sleep time is adequate. Sleeping at inconsistent times confuses the system. Dopamine cycles drift. The brain loses temporal coherence. This incoherence is often experienced as brain fog, low motivation, and emotional volatility. The person may feel out of sync with themselves. These states are not psychological quirks. They are neurochemical mismatches. The brain expects certain processes to occur at certain times. Hormone release, temperature regulation, neurotransmitter cycling, and gene expression are coordinated. When behavior conflicts with these rhythms, systems compete. Late-night stimulation keeps dopamine engaged when it should be settling. Morning light is delayed. The entire cycle shifts. The nervous system is adaptable. It can adjust to night work and irregular schedules, but this adaptation has a cost. Baseline arousal rises. Rest becomes less restorative. Dopamine signaling becomes less efficient. This is why shift work is associated with higher rates of mood disorders, metabolic disease, and cognitive impairment. It is not because night workers are weak. It is because their brains are constantly recalibrating. Sleep also modulates stress. Cortisol follows a circadian pattern. It rises before waking and falls through the day. Disrupted sleep blunts this rhythm. Cortisol remains elevated. Dopamine adapts defensively. The result is a nervous system that is both tired and alert. The body feels heavy. The mind feels restless. In this state, habits become harder to change. The prefrontal cortex is underpowered. The basal ganglia dominate. The system relies on routine. Motivation feels unreliable. Some days energy appears briefly, other days it vanishes. This unpredictability often leads to self-criticism. People assume their inconsistency reflects character. In reality, it reflects chemistry. Restoring rhythm is therefore foundational. It is not an accessory to change. It is the ground on which change occurs. Light is the primary cue. Morning light anchors the clock. Evening darkness allows melatonin to rise. Screens interfere because they emit wavelengths that the brain interprets as daytime. This does not mean technology must be eliminated. It means timing matters. The nervous system responds to patterns. Consistent wake times are more powerful than consistent bedtimes. The clock anchors to the morning. Evening sleepiness follows. Regular meals reinforce rhythm. Physical movement in daylight reinforces wakefulness. Quiet in the evening supports downshift. These are not lifestyle tips. They are signals to a biological system. When rhythm stabilizes, dopamine begins to cycle again. Motivation becomes more predictable. Energy returns in waves rather than spikes. This does not create constant drive. It creates reliable capacity. Sleep also restores emotional regulation. The amygdala becomes less reactive. The prefrontal cortex regains influence. The brain can evaluate rather than react. This shifts how dopamine is deployed. The system becomes less hijacked by threat. It becomes available for learning. People often try to change habits without changing sleep. They ask the system to learn while depriving it of the conditions required for learning. This is like asking a muscle to strengthen without rest. The system adapts by conserving. Sleep is therefore not separate from dopamine. It is the environment in which dopamine regains precision. The circadian system is not about productivity. It is about coherence. It aligns internal processes so that effort and rest alternate. When this alternation is lost, the nervous system becomes stuck in between. Not fully awake, not fully restored. The brain does not interpret this as inconvenience. It interprets it as instability. Dopamine in such a system becomes erratic. Some days it overfires. Some days it withdraws. The person feels unreliable to themselves. Restoring rhythm is not instant. The clock shifts slowly. The system resists abrupt change. Gradual consistency works better than dramatic adjustment. This is not discipline. It is entrainment. The brain learns through repetition. As rhythm returns, the reward map stabilizes. Actions begin to feel proportionate. Effort feels connected to outcome. This sets the stage for understanding how dopamine changes across the lifespan, because circadian rhythm, sleep architecture, and neurochemical balance evolve with age, reshaping motivation and experience over time. Dopamine does not remain the same throughout life. It changes in density, sensitivity, and pattern from infancy to old age. These changes are not cosmetic. They reshape how the world feels from the inside. They influence curiosity, risk-taking, patience, learning speed, emotional tone, and the way time itself is experienced. What feels natural at one stage of life can feel impossible at another, not because identity has vanished, but because the chemical landscape supporting experience has shifted. In early childhood, dopamine systems are extraordinarily plastic. The brain is built for rapid learning. Almost everything is new. Every sound, texture, face, and pattern carries information. Dopamine neurons respond vigorously to novelty. The world is saturated with prediction errors. Each day rewrites the map. This is why children appear endlessly curious. They are not exerting effort. Their brains are tuned to explore. The unknown is not threatening. It is expected. Dopamine rises easily. Attention is pulled outward. Learning feels natural. At the same time, inhibitory systems are immature. The prefrontal cortex develops slowly. the circuits that restrain impulse lag behind those that generate drive. This imbalance produces the characteristic energy of childhood. Action comes before reflection. Curiosity spills into movement. This is not recklessness, it is development. The brain is sampling the world at high speed. As adolescence approaches, dopamine systems undergo another shift. Receptor density changes, sensitivity increases in reward circuits, social cues become especially potent, novelty gains weight, the system becomes biased toward exploration and peer feedback. This is not a cultural artifact, it is a biological phase. The adolescent brain is tuned to leave the familiar and test boundaries. From an evolutionary perspective, this promotes dispersal and mating. It expands the gene pool. From the inside, it feels like intensity. Emotions sharpen, boredom deepens, rewards feel larger, risks feel less costly. The system overweights immediate outcomes and underweights long-term consequence. The prefrontal cortex continues to mature through this period. Its regulatory influence grows slowly. The result is a nervous system with strong drive and limited restraint. This is why adolescence is associated with experimentation. It is also why it is vulnerable. Substances and behaviors that strongly activate dopamine circuits can leave lasting traces. The map is still forming. Signals shape it deeply. This does not mean adolescence is fragile. It means it is formative. Experiences during this time carry disproportionate weight. As adulthood settles in, dopamine systems stabilize. Receptor density peaks and then gradually declines. The balance between drive and restraint evens out. Novelty becomes less intoxicating. The reward map becomes more refined. The brain now carries a history. Many patterns are known. Prediction errors become less frequent. The world feels more predictable. This is often experienced as loss of wonder. In reality, it is the result of a map that has been filled in. Fewer surprises mean fewer dopamine spikes. At the same time, prefrontal circuits are fully developed. Planning, patience, and perspective become easier. Goals can be sustained over time. The system supports delayed reward. This phase favors construction. Careers, relationships, and long-term projects become possible. The brain is tuned for maintenance and growth rather than exploration. Motivation during this period is shaped less by novelty and more by meaning. Dopamine still drives action, but it responds more strongly to goals that align with identity. The system becomes selective. This selectivity can feel like narrowing. Interests consolidate. The brain invests where it has learned value. Stress, however, can distort this balance. Chronic pressure can push the system back towards short-term relief. The brain adapts to threat. Dopamine shifts. Midlife often brings another change. Hormonal transitions interact with neurotransmitter systems. In some people, baseline dopamine declines. Energy patterns shift. Recovery takes longer. This is not decline in a simple sense. It is a reorganization. The brain becomes less responsive to novelty and more responsive to coherence. Chaos becomes costly. The system favors stability. This is often experienced as a desire for simplicity. Stimulation that once felt exciting now feels draining. The nervous system has less tolerance for noise. At the same time, emotional regulation often improves. The amygdala becomes less reactive. Perspective deepens. The brain weighs experiences differently. Dopamine in this phase becomes less about chasing and more about sustaining. Motivation shifts from expansion to preservation. This is not stagnation. It is adaptation to a different horizon. In later life, dopamine systems continue to change. Receptor density declines. Baseline levels decrease. The ability to generate strong anticipatory signals weakens. This can manifest as reduced drive. Tasks feel heavier. Initiation becomes slower. This is not apathy. It is a change in the chemistry of movement. At the same time, emotional tone often becomes more even. Many older adults report greater contentment. The The brain may rely more on systems associated with satisfaction than anticipation. The world feels slower, time feels different, the future carries less pull, the present becomes more central. This is not loss of ambition, it is a shift in orientation. Neurodegenerative diseases such as Parkinson's reveal the role of dopamine starkly. As nigrostriatal pathways deteriorate, movement becomes difficult. The mind remains willing. The body hesitates. This separation between intention and action illustrates how deeply dopamine is embedded in agency. Across the lifespan, then, dopamine reshapes what feels natural. Childhood favours exploration. Adolescence amplifies intensity. Adulthood supports construction. Later life favours stability. Each phase is adaptive. Each carries costs and gifts. Many struggles arise when life demands do not match the brain's phase. A young brain in a rigid environment feels trapped. An older brain in a chaotic environment feels overwhelmed. Understanding these shifts reduces self-blame. It reveals that motivation is not static. It is a moving target shaped by biology. This perspective also reframes generational differences. What feels obvious to one age group may feel alien to another. Their dopamine systems are tuned differently. Children seek novelty. Adults seek purpose. Elders seek coherence. These are not values, they are orientations. Life becomes easier when environments respect these orientations. Education thrives when it harnesses curiosity. Work thrives when it aligns with meaning. Aging thrives when it honours rhythm. Dopamine does not dictate destiny. It biases experience. It tilts the field. Awareness of these shifts allows compassion towards self and others. It explains why energy changes. It explains why interests evolve. It explains why what once motivated no longer does. This is not loss. It is movement through phases. And as technology increasingly shapes environments across all ages, these natural shifts encounter artificial stimuli that do not respect developmental timing. Understanding this interaction becomes essential. Technology did not invent dopamine, distraction, or desire. What it did was build an environment that speaks to these systems continuously, at scale, and with extraordinary precision. Every screen, every feed, every alert is a carefully shaped signal delivered directly into the nervous system. The brain that evolved to notice a rustle in the grass or the movement of another human now receives hundreds of such signals each hour, many of them designed to be unpredictable, emotionally charged and immediately available. The result is not a broken mind. It is a mind doing exactly what it has always done, responding to cues that promise change. The infinite scroll is one of the clearest examples of how ancient learning circuits meet modern design. In the physical world, exploration has edges. A forest ends. A path turns. A book has a final page. Each boundary provides closure. The brain reaches an endpoint and the dopamine-driven anticipation cycle resolves. The system rests. The infinite scroll removes that boundary. There is no natural stopping point. Each movement of the thumb reveals something new. The brain never receives the signal that the search is complete. Anticipation remains active. The reward map stays engaged. This is not an accident. Variable reward schedules are known to produce strong learning. They keep organisms engaged because each interaction might produce something meaningful. Slot machines use this principle. So do social feeds. Most of what appears on a feed is neutral. Occasionally something resonates. The unpredictability is what matters. The dopamine system cannot predict which swipe will matter. It remains alert. The nervous system experiences this as light tension, a subtle readiness. The mind leans forward. It is not unpleasant. It is engaging. Over time, this engagement becomes baseline. The brain begins to expect frequent novelty. Quiet moments feel empty. The absence of input feels like deprivation. This is not addiction in the clinical sense for most people. It is adaptation. The dopamine system recalibrates based on environment. When signals arrive constantly, the baseline shifts. What once felt stimulating now feels ordinary. The system seeks more. This does not require hours of use. It occurs through repetition. Each interaction reinforces the pattern. Technology also compresses time. In natural environments, effort and reward are separated by distance and delay. You walk to a place, you build a tool, you learn a skill. Dopamine supports the bridge across that gap. In digital environments, the gap collapses. Reward arrives instantly. The system learns that effort is unnecessary. The bridge shortens. This changes how the brain evaluates tasks. Activities that require sustained effort without immediate feedback feel heavier. Reading deeply, practicing a skill, writing. These tasks no longer match the trained rhythm. The brain does not interpret this as challenge. It interprets it as inefficiency. This is not because the person lacks discipline. It is because their nervous system has learned a different pattern of engagement. Attention itself is shaped by this. The brain becomes adept at rapid shifting. It learns to sample. It becomes less comfortable with stillness. The prefrontal cortex must work harder to maintain focus. That work consumes energy. Fatigue sets in more quickly. Many people experience this as reduced patience. They feel restless. They struggle to stay with one thing. This is not a failure of character. It is a learning outcome. Technology also amplifies social dopamine. Approval becomes quantifiable. Belonging becomes visible. The brain learns to monitor. Each notification is a cue. Each cue triggers anticipation. The system checks. The loop continues. In face-to-face life, social feedback is slower and richer. Online, it is compressed and abstract. The brain fills in gaps. This can heighten sensitivity. Absence of response feels like rejection. The nervous system remains vigilant. The infinite scroll does not only deliver content, it delivers potential. Each swipe might reveal something that matters. The brain cannot easily disengage from potential. In physical exploration, the body tires. In digital exploration, the body remains still. The system does not receive signals of exhaustion. The loop continues beyond natural limits. This produces a peculiar state. The mind is stimulated. The body is inert. The nervous system remains in a low-level anticipatory mode. Over time, this state becomes familiar. The brain learns to associate rest with input. Stillness becomes uncomfortable. This alters the meaning of boredom. Boredom once signaled that the environment lacked stimulation. It motivated exploration. In digital environments, boredom becomes a momentary glitch. It is immediately resolved by a swipe. The brain never learns to sit with low stimulation. It never practices internal generation of interest. Curiosity becomes externally driven. This does not eliminate creativity. It changes its source. Technology is not inherently harmful. It expands access to information, connection, and expression. It enables learning at scale. It allows ideas to travel. The issue is not presence, it is proportion. The dopamine system evolved for environments with scarcity of novelty. It now lives in abundance. It is not calibrated for endless signal. The nervous system adapts by raising thresholds. What once felt engaging becomes baseline. What once felt satisfying becomes brief. This can create a sense of dullness. People report feeling numb. They seek stronger input. This is not because life lacks meaning. It is because the system has been trained to expect high-frequency change. Recovery does not require rejection of technology. It requires restoration of boundaries. Boundaries allow loops to close. They allow anticipation to resolve. They allow the system to rest. A book ends. A walk returns. A task completes. These experiences teach the brain that effort leads to completion. Without such experiences, the map remains open-ended. The nervous system is not offended by technology. It is impressionable. Introducing finite activities recalibrates. Reading a chapter, cooking a meal, completing a puzzle. These provide closure. Silence is not emptiness. It is a signal that no action is required. It allows cortisol to fall. It allows dopamine to downshift. Initially, silence may feel uncomfortable. The brain has learned to expect input. This discomfort is not a verdict. It is withdrawal from constant stimulation. Over time, quiet becomes neutral again, then restorative. The infinite scroll is powerful because it aligns perfectly with the dopamine system's bias toward anticipation. It is not a trap. It is a mirror of ancient circuitry. Understanding this reduces shame. It reveals that engagement is not a personal flaw. It is a biological response. Choice becomes possible when pattern becomes visible. This understanding naturally leads into how balance can be reclaimed, not by force, but by reshaping the learning environment. Balance is often imagined as something achieved through discipline, a state reached by forcing the mind into better habits. In reality, balance emerges from alignment between environment and biology. The nervous system does not respond to moral instruction. It responds to patterns. Dopamine does not listen to intention. It adapts to experience. Reclaiming balance in a high-stimulation world is therefore not about resisting desire. It is about reshaping the learning landscape so that desire can settle into rhythms that match the brain's design. The first shift is conceptual. Stimulation is not the enemy. The brain is built to seek. Curiosity, novelty, and reward are essential. The problem arises when these signals arrive without boundary or resolution. The system remains engaged without rest. Balance does not mean eliminating stimulation. It means restoring contrast. Contrast is what gives experience shape. Without contrast, everything flattens. When every moment carries signal, no moment stands out. Dopamine relies on difference. It responds to change. When change is constant, change loses meaning. The nervous system needs periods where nothing is demanded. Not because productivity is wrong, but because recalibration requires quiet. This quiet is not boredom in the pejorative sense. It is low stimulation. It is a state where the brain is not being pulled forward by cues. Initially, this state feels uncomfortable. The system has learned that something is always coming. When nothing arrives, the brain interprets it as absence. The feeling is restlessness. This restlessness is not evidence that quiet is harmful. It is evidence of adaptation. The brain is plastic. It learns through repetition. If low stimulation is encountered regularly and does not lead to harm, the system updates. The discomfort fades. Quiet becomes neutral. This process mirrors habit formation. Each time a cue fails to produce stimulation, a small prediction error occurs. Dopamine dips. Over time, the association weakens. This is not deprivation. It is retraining. The same principle applies to attention. Focus is not a trait. It is a skill supported by chemistry. The brain becomes better at what it practices. If attention is constantly fragmented, the circuits that support sustained focus weaken. If attention is held, even briefly, those circuits strengthen. This does not require hours of concentration. It requires repeated moments of staying. Reading a page without switching. Listening to a song without multitasking. Walking without a device. These moments train the system. Each time attention remains, a different loop closes. Dopamine learns that engagement can persist without constant novelty. The goal is not to suppress curiosity. It is to allow it to deepen. Curiosity that hops is not wrong. It is shallow. Curiosity that stays becomes understanding. The brain can learn both. Environment shapes this more than will. Removing cues reduces activation. Placing devices out of reach changes behavior without effort. Creating spaces where stimulation is absent gives the system room. This is not about austerity. It is about design. The nervous system is not a rational agent. It is a learning machine. It adapts to what is present. A home where every surface glows trains a different brain than one where light fades in the evening. These changes do not need to be absolute. They need to be consistent. Consistency teaches the system what to expect. Morning light anchors rhythm. Evening dimness signals rest. Regular meals provide temporal markers. Physical movement creates embodied feedback. These patterns restore coherence. Coherence is what allows dopamine to function precisely. It thrives in environments where signals have meaning. When everything is signal, nothing is. Reclaiming balance also involves restoring arcs. Activities with beginnings and endings teach the brain closure. Cooking, gardening, writing a letter. These tasks have natural limits. The nervous system recognizes completion. Dopamine subsides. Satisfaction emerges. This satisfaction is quieter than stimulation. It does not spike, it settles. At first it may feel thin. The brain is accustomed to intensity. Over time it gains depth. The difference between stimulation and satisfaction is subtle but important. Stimulation excites. Satisfaction grounds. The modern environment offers abundant stimulation and little grounding. Rebalancing does not require rejecting modern life. It requires adding grounding. This can be as simple as creating device-free intervals, not as punishment, but as experiment. The brain needs evidence that nothing breaks when stimulation pauses. During these intervals, the mind may wander. It may seek. It may feel uneasy. This is normal. Over time, the system learns that safety persists. Stress systems downshift. Dopamine cycles soften. Balance also involves acknowledging limits. The nervous system is not designed for constant input. Fatigue is not failure. It is signal. Rest is not a reward. It is maintenance. In a high-stimulation world, rest must be protected, not earned. This protection can be structural. Fixed bedtime, morning routines, boundaries around work. These are not lifestyle hacks. They are biological supports. The brain does not care about productivity. It cares about predictability. Predictability reduces cortisol. Lower cortisol restores dopamine sensitivity. The system becomes more responsive. Motivation returns not as urgency, but as readiness. This readiness feels different. It is quieter. It is steadier. It allows action without compulsion. The same principle applies to goals. Large goals overwhelm the system. They create distant prediction errors. Dopamine does not bridge easily. Breaking goals into visible steps provides frequent feedback. Each step closes a loop. The system learns that effort works. This is not trickery. It is alignment with learning. Balance is not stasis. It is rhythm. The nervous system expects cycles, effort and rest, seeking and settling, engagement and withdrawal. When these cycles are disrupted, the system becomes noisy. Reclaiming balance is restoring cycle. It does not require perfection. It requires repetition. Small, consistent changes shape chemistry. The brain is patient. It does not change in a day, it changes in patterns. Understanding dopamine reveals that change is not heroic. It is incremental. The system does not respond to declarations, it responds to experience. Each moment of low stimulation is a lesson. Each completed task is a lesson. Each night of sleep is a lesson. Over time, the reward map changes. The world regains depth. This does not eliminate desire, it refines it. Desire becomes selective, it aligns with meaning. Balance, then, is not restraint. It is resonance. It is living in a way that allows the nervous system to function as designed. This leads naturally into the final reflection, where the question is not how to control dopamine, but how to live gently with it, recognizing that it is not a master to defeat, but a companion that can be understood. To live with a human nervous system is to live inside a moving system of signals. Every thought, Every urge, every pause arises from chemistry in motion. Dopamine is only one part of that motion, but it sits at a crucial intersection between what is and what might be. It does not tell you what to value. It tells your brain what seems worth moving toward. Much of life feels personal because it is experienced from within. Desire feels like intention. Restlessness feels like personality. Motivation feels like character. But beneath these experiences lies a system that evolved long before language, before culture, before self-reflection. It was built to keep organisms moving through uncertain environments, not to make them content. Understanding this does not reduce meaning, it relocates it. It reveals that struggle is not always a failure of will. It is often a mismatch between ancient circuitry and modern conditions. Dopamine does not make you shallow when you reach for stimulation. It does not make you weak when habits persist. It does not make you broken when motivation fades. It is responding to patterns it was designed to learn. Living gently with your chemistry begins with this recognition. You're not an abstract mind riding a flawed body. You're an embodied system shaped by signals. Your inner life is not separate from molecules. It is what those molecules feel like from the inside. Gentleness does not mean indulgence, it means accuracy. It means responding to what is actually happening rather than to what you think should be happening. If attention wanders, it is not because you lack depth. It is because your environment is rich in cues. If effort feels heavy, it is not because you are lazy. It is because the bridge between effort and reward has weakened. If rest feels uncomfortable, it is not because you are incapable of stillness. It is because your system has learned that stimulation is constant. None of these states are permanent. They are learned. What is learned can change. But change in a biological system does not happen through argument. It happens through repetition, through exposure, through pattern. Living gently with your chemistry means working with that reality. It means choosing environments that teach the nervous system what you want it to know. It means giving your brain experiences that allow it to update. This is not self-optimization. It is self-alignment. You cannot command dopamine. You can shape what it encounters. You cannot force motivation. You can provide conditions where motivation becomes possible. You cannot will yourself into balance. You can create rhythms that allow balance to emerge. This begins with noticing. Notice when anticipation pulls you forward. Notice when quiet feels uneasy. Notice when small tasks feel heavy. These are not failures, they are signals. Signals do not require judgment, they require interpretation. A body that is hungry is not flawed, it needs nourishment. A nervous system that is overstimulated is not weak, it needs quiet. A mind that resists effort is not broken, it may be depleted. Living gently means responding to these needs without shame. Shame adds noise, it activates stress, it distorts dopamine further. It teaches the system that inner experience itself is threat. Gentleness teaches safety. Safety is the condition under which learning occurs. The brain updates when it is not defending. This is why compassion is not a soft concept. It is a biological one. It lowers cortisol, it restores prefrontal function, it allows dopamine to regain precision. When you treat your inner state as an enemy, your nervous system prepares for conflict. When you treat it as information, it becomes adaptable. This does not mean every impulse should be followed. It means every impulse should be understood. Dopamine will continue to rise in response to novelty. It will continue to mark cues. It will continue to lean you forward. That is its nature. Living gently does not require stopping that motion. It requires giving it places to land. It requires allowing anticipation to resolve. Completion is kind to the nervous system. So is rest. So is rhythm. So is meaning. Meaning is not abstract to the brain. It is encoded in reliability. When actions lead to outcomes that matter, dopamine aligns with purpose. When actions lead nowhere, the system withdraws. You do not need to find grand meaning. Small coherence is enough. A walk that ends, a page that finishes, a meal that is cooked, a thought that is written. These are anchors. They teach the brain that effort connects to reality. Over time, these experiences rebuild trust. Trust is not a belief. It is a chemical expectation. A nervous system that trusts its environment invests. A nervous system that distrusts conserves. Living gently is building trust with your own biology. It is letting the system experience that nothing bad happens when you slow down. It is letting it experience that some things are worth staying with. It is letting it experience that rest does not mean abandonment. These lessons cannot be explained. They must be felt. They are learned in quiet moments, in repeated routines, in small closures. Dopamine will still rise. Desire will still appear. Curiosity will still pull. The difference is that it will no longer dominate. It will become one voice among others. Satisfaction will have space. Presence will have weight. The present moment will no longer feel like a waiting room. This is not a promise of constant peace. It is a promise of coherence. Life will still include stress. It will still include uncertainty. The nervous system will still respond, but it will have reference points. It will know where to return. Gentleness is not passive. It is deliberate. It is choosing to work with a system rather than against it. It is recognizing that you are not the author of your chemistry, but you are its environment. Every choice of rhythm, light, silence, movement, and focus teaches your brain something. You are always training it. The question is not whether dopamine controls your life. The question is whether you understand the language it speaks. When you do, you can stop arguing with your own biology. You can stop mistaking signals for flaws. You can begin to live in a way that respects the machinery that makes experience possible. And in that respect, the nervous system often softens. The chase slows. The present gains texture. Rest becomes allowed. Not because you have mastered yourself, but because you have learned how to listen. And listening for a biological system is the beginning of change. As this session comes to a close, there is nothing here that needs to be remembered, mastered, or acted upon. Everything you've heard is already happening inside you, quietly, automatically, without effort. The chemistry that moves through your brain tonight will continue whether you think about it or not. Dopamine will rise and fall, signals will travel, circuits will shift. Your nervous system will keep doing what it has always done, adapting. The difference now is simply that these processes are no longer invisible. They have shape, they have logic, they have history. What once felt like personal struggle can be understood as biology meeting circumstance. That understanding does not solve everything, it softens it. There's no need to change anything right now. The brain does not rewire in response to insight alone. It rewires through time, repetition, and rest. Even listening is already part of that process. Attention has slowed. Stimulation has eased. The system has been allowed to drift. This is how recalibration begins, not through force, but through quiet exposure to a different rhythm. Wherever you are listening from, your body is already taking over. Breathing continues. Muscles release. Thought thins. The same chemistry that carried you through the day is now guiding you toward rest. You do not have to manage it. You do not have to guide it. You can let it work. If this space has been helpful, you can return to it whenever you need a place where learning does not demand performance. A place where science is allowed to be slow. A place where your nervous system does not have to prove anything. For now, the only task is to remain where you are, doing nothing at all. The brain knows what comes next.